Hey biology students, we're now going to do our second video on pathogenic bacteria. This is continuing from the first video, and I'm calling this lecture video Patterns of Infectious Disease. In this figure, we're looking at the progression of disease, infectious disease, using some disease terminology that you should become familiar with. Whoops. Okay, so the initial exposure. So we're looking at a graph. We're looking at time with no specific numbers because this is a highly variable situation. In fact, not all infectious diseases play out like this. So this is a really generalized diagram. So we're looking at a generalized time progression on the x-axis, and then on the y-axis, we're looking at intensity of symptoms. That also correlates to the number of microbes. So that can also be here. So number of microbes would also be increasing as we move up on the y-axis. And this also relates to the immune response to that specific pathogen. That immune response also plays out with this story here. So here it's showing us the initial exposure, time zero, initial exposure. And so from that time you become exposed to the pathogen, there is what we call the first phase, and that is called the incubation period. Most of the time, the incubation period is asymptomatic. So the person has no symptoms of the infection. And this can last for a variable amount of time. Hours, days, months. It varies depending on the pathogen we're talking about, okay? Following that incubation period, notice everything is really low. Numbers of microbes are low, immune response is low, and there's really no symptoms that are showing up. Second stage, prodromal stage, and then that exponentially increases into what we call the invasive phase. And so now we begin to see increase in these numbers of microbes. Recall, we actually did a graph that was really similar to this in the beginning of the semester regarding growth curves of bacteria. Do you remember? So the incubation period actually corresponds to what we call the lag phase in the growth of bacteria, where there's just a few cells that are introduced into the body and they are acquiring nutrients, but they haven't quite started their exponential growth phase. Okay, remember that exponential growth phase is called the log phase. So this terminology, we can kind of overlay it with the disease terminology as well. So log means exponential growth. So eventually the bacteria, they become established and they begin to grow and double and double and double. And then we get large quantities of these microbes, and we hit our peak of infection. And of course, what's happening? The symptoms, right? Symptoms are going up. Patient is becoming symptomatic. And like I said, this is a really generic curve because there are infections that are completely asymptomatic, even in that invasive phase. So this isn't a perfect 100% of the time type of graph, okay? Eventually what happens is hopefully this is going to be a happy story, okay? And the happy story is that the body overcomes the infection, and that's because of the immune response. So the immune response reaches its peak, and that's because of the activation of adaptive immunity. Okay, eventually the T cells and the B cells become activated, and those guys are the key players in wiping out an infection. If they become activated to high levels, then we can get rid of that specific pathogen. And then we enter the convalescent period, where we begin to see a decrease in the number of microbes. We see a suppression of the immune response as everything is cleared, and we see those symptoms start to go away. When is a person contagious? Well, in general, we say the 4 stages, all of those stages, to a lesser degree during incubation and convalescence. but it is possible. In fact, this is one of the reasons why we are seeing such huge numbers of COVID-19. It's because people with COVID-19 are asymptomatic during the incubation period, but also highly contagious. And so they may not, what we mean again by incubation is that this is the time from when they were initially exposed and then to when they actually show the first symptoms, runny nose, cough, fever, shortness of breath, somewhere along those lines. Um, they will not have symptoms in here. And so that is going to cause them to go out about their lives normally and spread the virus. And we're seeing lots of asymptomatic spread of this virus, which is why the quarantine and the basically stopping of all normal life where we all have this shelter in place order, why that hopefully is going to be effective. It's going to keep those asymptomatic people in the incubation period from spreading the virus. I also want to draw your attention to terminology related to infectious disease that varies. There's a difference between what we call signs and symptoms of an infection. So everything on the left is a sign. What does that mean? It's something that you can observe or measure. So if you are not the person who's actually sick and you're a caretaker for that person, you can measure their fever. We can measure, this is septicemia, the amount of bacteria in their blood or viral particles in their blood. We can measure their chest sounds. Leukocytosis refers to the number of leukocytes. So we can take a blood sample. We could see what kind of leukocyte number we have. Usually that refers to an increase in leukocytes, or leukopenia refers to a decrease in leukocytes. Okay, so everything on the left we can observe and measure. Symptoms, this refers to what the patient experiences, which is highly subjective to the patient. It's not something we can necessarily measure, although there are ways you can try to measure this where you could ask the patient, for example, everybody experiences pain very differently. And so you can actually ask the patient, you could say on a scale of 1 to 10, how would you rate your pain? They did that a lot to me when I was in labor to try to get an idea, right, of when you're going to need that epidural. So, we can try to quantify it, but in general, this is all very subjective to how the patient feels. But when we talk about an infectious disease, it's a combination of these two things. It's the signs that we can measure, and then it's the symptoms as well that the patient experiences. Infectious diseases follow this generic pattern. So it begins where we have introduction of the pathogen into the body, and we call that a portal of entry. How does it get in? How does the pathogen get into the body? Well, various ways. Through the skin, breaking that first line of defense, these barriers. Gets in through the GI tract. So from what we're eating, okay, into the mouth, into the stomach and intestines, right? So food or water we ingest. The respiratory tract. So that includes the nose and the mouth. The urogenital tract. So the genitals, including the urethra as well. And then what happens? They attach firmly. So once those microbes get in, they need to make sure they're not flushed out, because remember, the body has flushing mechanisms. There's mucus layers, there's stomach acid, there's just the process of urination that's continuously flushing our body. And so in order for that pathogen to establish an infectious disease, it needs to be able to attach And so bacteria have certain strategies we've discussed before, like those little fimbriae hairs that they use, or they might use their glycocalyx, that sugary coating that certain bacteria have. For viruses, it's more often their spike proteins, their surface proteins that helps them adhere to cells. So once you attach yourself as a pathogen, the pathogen has to survive the host defenses. It's probably already gotten past the first line of defense. Actually, it's definitely gone past the first line of defense. But the second line of defense will now come and try to attack, and those are going to be our leukocytes. And so the leukocytes are going to notice, okay, that we've got invaders, and so they need to survive the host defenses or have mechanisms. Successful pathogens have mechanisms for surviving the host defenses. So this is highly variable what they do. My next slide, we'll go to that now, actually gives you some ideas of just some examples of various pathogens that have mechanisms for surviving their host defenses. So these are just four that we can consider. So Staphylococcus aureus produces a toxin that just goes and straight out kills the leukocyte. Well, there you go. That's one way to prevent phagocytosis. Streptococcus pneumoniae, the cause of bacterial pneumonia, has a antiphagocytic capsule around it. We've talked about that particular virulence factor before, and that prevents phagocytosis. What about Mycobacterium tuberculosis? So when Mycobacterium tuberculosis is phagocytosed into the phagosome, it actually prevents the formation of the phagolysosome. So that prevents digestion. This is how the tuberculosis bacteria replicates. It does so inside of a macrophage. And so does Listeria. So Listeria monocytogenes, another pathogen that actually disrupts the phagolysosome. It does so by actually digesting it and breaking it down, and then that releases the Listeria bacteria into the cytoplasm of the macrophage where it can then replicate. So these are some of the classic ways. There's also another very classic way of surviving host defenses, and it's called antigenic variation. And this is a result of mutation. When pathogens mutate, those pathogens that have a high mutation rate, that's actually an advantage for the pathogen because it changes the surface antigens. And we've talked about this before, but this is like changing your appearance. So if you have a criminal that everybody's searching for and they have a particular physical appearance, but then that criminal cuts their hair or they dye their hair or they change their appearance, and now they look significantly different to the police. And the same thing can happen here with the pathogens, is they can change their surface proteins. For example, influenza virus is very good at doing this, which is why the flu vaccine is only effective for about 1 year. And you need to get the flu vaccine every year in order to have complete protection against the influenza virus, because it's very good at mutating and changing those surface antigens. And recall, that's how the immune system works. It's a memory response. And so if that pathogen looks different, has different antigens, then those memory cells are not going to remember it. And we're going to have to start all at square one again with going through antigen presentation and finding and activating the T and the B cells that recognize that specific pathogen. Okay, so survive the host defenses. Step 4, cause damage or disease. And they do this in actually sort of 2 different strategies for causing damage or disease. One is going to be to do direct damage to the host. And this is usually accomplished from various toxins. We're going to talk about toxins next, or enzymes that are produced by the bacteria. So we can get direct damage, so the cells of the body actually do get directly damaged, or the other popular approach of the pathogen is indirect damage. And what that means is the immune system is overstimulated. So the immune system is overreactive. We've talked about this in relationship to our current pandemic of COVID-19 as well, that the overactive inflammatory response is what is contributing to the viral pneumonia. And so the pathogen is facilitating that. Okay. Step 5, exit the host. Now, this is really important for the pathogen to do this because if the host dies early on, then that pathogen probably isn't going to be transmitted. So the pathogen needs to make sure that it can actually exit because it needs to find a new host to survive in. And so how's it going to exit? Well, pretty much the same way it got in. So it's going to exit either through the skin, it's going to exit through respiratory secretions like coughing and sneezing. Oops. Fecal fecal matter, so or urine, blood, body fluids. Okay, so it's going to get out and then it's going to transmit, and the cycle repeats to a healthy host.